Chapter 1, Muscle Physiology. This chapter provides an overview of how muscles work to create forces across joints and cause movement. Understanding the physiology of muscle is basic to developing and implementing training programs. At the simplest level, the strength and conditioning professional is concerned with the function of the muscle. Many systems, including the cardiovascular, nervous, and endocrine systems, affect the function of muscle. But muscle is the organ that creates movement. In other words, the other systems affect performance by changing how the muscle functions. Skeletal muscle, macrostructure, and microstructure. Each skeletal muscle is an organ that contains muscle tissue, connective tissue, nerves, and blood. Fibrous connective tissue, or epimysium, covers the body, more than 430 skeletal muscles. The epimysium is continuous with the tendons at the ends of the muscle. The tendon is attached to the bone periosteum, a specialized connective tissue covering all bones. Any contraction of the muscle pulls on the tendon, and in turn the bone. Limb muscles have two attachments to bone: proximal, closer to the trunk, and distal, further from the trunk. The two attachments of trunk muscles are termed superior, closer to the head, and inferior, closer to the feet. Traditionally, the origin of the muscle is defined by its proximal, towards the center of the body attachment, and its insertion is defined as its distal, away from the center of the body attachment. Muscle cells, often called muscle fibers, are long, sometimes running the entire length of a muscle. Cylindrical cells, 50 to 100 micrometers in diameter, about the diameter of a human hair. These fibers have many nuclei situated on the periphery of the cell and have a striated appearance under low magnification. Under the epimysium, the muscle fibers are grouped into bundles, fasciculi, that may consist of up to 150 fibers, with the bundles surrounded by connective tissue called perimysium. Each muscle fiber is surrounded by connective tissue called endomysium, which is encircled and continuous with the fibrous membrane, or sarcolemma. All the connective tissue, epimysium, perimysium, and endomysium, is continuous with the tendon, so tendon developed in one muscle cell is transmitted to the tendon. The junction between a motor neuron, nerve, and cell, and the muscle fiber it innervates is called the motor end plate, or more often, the neuromuscular junction. Each muscle cell has only one neuromuscular junction, although a single motor neuron innervates many muscle fibers, sometimes as many as several hundred. A motor neuron of the muscle fiber it innervates and the muscle fiber it innervates are called a motor unit. All the muscle fibers of a motor unit contract together when they are stimulated by the motor neuron. The interior structure of a motor fiber is depicted in figure 1.3. The sarcoplasm, the cytoplasm of a muscle fiber, contains contractile components, which consist of protein filaments, other proteins, stored glycogen, and fat particles, enzymes, and specialized organelles such as mitochondria and the sarcoplasmic reticulum. Hundreds to thousands of myofibrils, each about 1 micrometer in diameter, 1/100th the diameter of a hair, dominate the sarcoplasm. The myofibrils contain the apparatus that contracts the muscle cell, which primarily of the two types of myofilament, myosin and actin. The myosin filaments, thick filaments, contain up to 200 myosin molecules. Globular heads called cross bridges protrude away from the myosin filament at regular intervals. The actin filaments consist of two strands arranged in a double helix. Myosin and actin filaments are organized longitudinally in the smallest contractile unit of skeletal muscle, the sarcomere. Sarcomeres in a relaxed fiber approximately 4,500 per centimeter of muscle length, are repeated the entire length of the muscle fiber. Figure 1.4 shows the structure and orientation of a myosin and actin in the sarcomere. Adjacent myosin filaments anchor to each other at the M bridge in the center of the sarcomere, the center of the H zone. Actin filaments are aligned at both ends of the sarcomere and are anchored at the Z line. Z lines are repeated through the entire myofibril. 6 actin filaments surround each myosin filament, and each actin filament is surrounded by 3 myosin filaments. It is the arrangement of the myosin and actin filaments and the Z lines of the sarcomere that gives skeletal muscle its alternating dark and light pattern, its striated appearance under magnification. The dark A band corresponds with the alignment of the myosin filaments, whereas the light I band corresponds with the areas in the 2 adjacent sarcomeres that contain only the actin filaments. The Z line is in the middle of the I band and appears as a thin, dark line running longitudinally through the I band. The H zone is in the area at the center of the sarcomere where only myosin filaments are present. During muscle contraction, the H zone decreases as the actin slides over the myosin towards the center of the sarcomere. The I band also decreases as the Z lines are pulled toward the center of the sarcomere. Parallel to and surrounding each myofibril is an intricate system of tubules called the sarcoplasmic reticulum that terminates as vesicles in the vicinity of the Z line. Calcium ions are stored in these vesicles. The regulation of calcium controls muscular contraction. T tubules, short for transverse tubules, run perpendicular to the sarcoplasmic reticulum and terminate in the vicinity of the Z line between the 2 vesicles. This pattern of a T tubule spaced between the perpendicular to 2 sarcoplasmic reticulum vesicles is called a triad. Because the T tubules run between outlining myofibrils and are continuous with the sarcolemma at the surface of the cell, discharge of an action potential, an electrical nerve impulse, arrives nearly simultaneously from the surface to all depths of the muscle fiber. Calcium is thus released throughout the muscle, producing a coordinated contraction. Sliding filament theory of muscular contraction. In its simplest form, the sliding filament theory states that the actin filaments at each end of the sarcomere slide inward on myosin filaments, pulling the Z line towards the center of the sarcomere, thus shortening the muscle fiber. As actin filaments slide over myosin filaments, both the H zone and I bands shrink. The flexion of myosin cross bridges pulling on the actin filaments is responsible for the movement of the actin filament. Because only a small displacement of the actin filament occurs with each flexion of a myosin cross bridge, very rapid repeated flexion must occur in many cross bridges throughout the entire muscle for the measurable movement to occur. Resting phase. Under normal resting conditions, little calcium is present in the myofibril. Most of it is stored in the sarcoplasmic reticulum. So very few of the myosin cross bridges are bound to actin. No tension is developed in the muscle, so the muscle is said to be at rest. Excitation, contraction, coupling phase. Before myosin cross bridges can flex, they must first attach to the actin filament. When the sarcoplasmic reticulum is stimulated to release calcium ions, the calcium binds with troponin, a protein situated at regular intervals along the actin filament that has high affinity for calcium ions. This causes a shift to occur in another protein molecule, tropomyosin, which runs along the length of the actin filament in the groove of the double helix. The myosin cross-bridge head now attaches more rapidly to the actin filament, allowing cross-bridge flexion to occur. Contraction phase. The energy for cross-bridge flexion comes from hydrolysis, breakdown of adenosine triphosphate, ATP, to adenosine diphosphate, ADP, and phosphate, a reaction catalyzed by the enzyme myosin ATPase. Another molecule of ATP must replace the ADP on the myosin cross-bridge head for the head to detach from the active actin site and recock. This allows the contraction process to be continued if calcium is available to bind to the troponin, or relaxation to occur if calcium is not available. It may be noted that calcium plays a role in regulating a large number of events in skeletal muscle, besides contraction. These include glycolytic and oxidative energy metabolism, as well as protein synthesis and degradation. Recharge phase. Measurable muscle shortening transpires only when the sequence of events binding calcium to troponin, coupling of the myosin cross-bridge with actin, cross-bridge flexion, dissociation of actin and myosin, and recocking of the myosin cross-bridge head is repeated over and over again throughout the muscle fiber. This occurs as long as calcium is available in the myofibril, ATP is available to assist in the uncoupling of myosin from the actin, and sufficient active myosin ATPase is available for catalyzing the breakdown of ATP. Relaxation phase. Relaxation occurs when the stimulation of motor nerve stops. Calcium is pumped back into the sarcoplasmic reticulum, which prevents the link between the actin and myosin filaments. Relaxation is brought about by the return of actin and myosin filaments to their unbound state. Fiber types. Not all muscle fibers are homogeneous regarding metabolic and contractile capabilities. Two different muscle fiber types have been identified: type 1, slow twitch, or type 2, or fast twitch. Briefly, type 1 muscle fibers resist fatigue by having a large number of mitochondria, high aerobic enzyme activity, and dense capillary concentration. Two main subdivisions of type 2 have been identified, although some investigation have identified additional subcategories. Type 2b, or fast glycolytic muscle fibers, fatigues rapidly and has relatively few mitochondria, low aerobic enzyme activity, and few capillaries. Type 2a, or fast twitch oxidative glycolytic muscle fibers, has some of the fatigue-resistant capabilities of type 1 muscle fibers, with more mitochondria, greater aerobic enzyme activity, and greater capillary density than type 2b muscle fibers. Type of muscle action. Concentric muscle action occurs when the total tension developed in all the cross bridges of a muscle fiber is sufficient to overcome any resistance to shortening. During the upward phase of a biceps curl, for example, the cross bridges in the biceps brachii and other elbow flexors overcomes the resistance of the barbell, arm, and hand. Isometric muscle actions occur when the tension of the cross bridge equals the resistance of shortening and the muscle length remains relatively consistent. This would occur during a biceps curl. Movement was stopped and the force developed in the elbow balanced the resistance of the barbell, arm, and hand. Eccentric muscle action occurs when the tension developed in the cross bridges is less than the external resistance, and the muscle lengthens despite contact between myosin cross-bridge heads and the actin filament. During the slow, controlled downward phase of a biceps curl, the cross bridges within the elbow flexors exert force to slow the descent of the barbell, but insufficient force to stop the descent or raise of the barbell. Force production. The number of myosin cross bridges that are attached to the actin filament at any instant in time dictates how much force is being produced in that muscle. A thorough understanding of factors that affect force production is necessary for developing conditioning programs that optimize improvements in sport-specific performance. Motor unit recruitment. A greater amount of calcium in a myofiber results in more myosin cross-bridge heads binding with actin filaments, and thus more tension in the muscle. The amount of calcium released from the sarcoplasmic reticulum vesicles is related to how frequently the innervating motor neuron stimulates the muscle. Increased frequency of stimulation of a motor unit results in increased force production of that motor unit. The number of active motor units can also affect force production. The greater the number of active motor neurons, the greater force produced. In short, force production is controlled in two main ways: frequency of stimulation of motor units and number of motor units activated. It is possible that some training-induced increases in strength may occur because of increased neural activity of motor units. Preloading. Although the release of calcium ions binding to myosin to actin occurs very rapidly, the process does not occur instantaneously in all the motor units or at all the myosin crossbridge heads. It takes some time for all the potential myosin crossbridge heads to make contact with the actin filaments. This means that maximal force production in a muscle does not occur instantaneously. Consequently, maximal force production may not occur early in the range of motion, especially during fast movements. Because high force development may be important in strength development, development of strength may be retarded under training conditions that do not allow high tension development in the early part of the range of motion. High tension is developed in a muscle even before movement occurs when lifting weights because the weight must be supported isometrically. This is called preloading. In addition, no movement can occur until sufficient force is developed in the muscle to overcome the inertia of the barbell. Preloading and inertia ensure high tension development early in the range of motion. Accommodating resistance apparatus such as hydraulic and isokinetic systems do not load the muscle prior to contraction. Research indicates that preloading during training may be important in development of strength early in the range of motion, especially at high velocities. Cross-sectional area. The maximum force capability of a muscle is related to the cross-sectional area of the muscle. Muscles that have larger cross-sectional area have larger number of sarcomeres in parallel, more potential cross-bridge heads in contact with actin molecules, and greater potential for applying force. In other words, thicker muscles have greater potential for generating force. Velocity of shortening. On the other hand, more sarcomeres in series, thus the longer muscle, increases the potential velocity of shortening. This may possibly occur because of the sarcomere shortening at about the same time. Two sarcomeres in series could shorten the ends of the muscle 2 times as far as the same amount of time as only one sarcomere. Larger muscles with more sarcomeres in series have greater potential for fast contractions. In addition, force production is inversely related to velocity of shortening during concentric actions. In other words, during fast movements, less force production is possible, and when lifting heavier loads, slower movement occurs. This is probably due to smaller number of cross-bridge contacts of actin filaments at any instant as the velocity of shortening increases. The relationship is different for eccentric actions. As the velocity of eccentric actions increases, maximum force production also increases. The force capabilities are typically 120 to 160% greater in eccentric actions than in concentric actions. This means that when overloading eccentrically, very heavy resistance may be needed, but when training for explosive concentric movements, relatively light resistance may be more suitable. Angle of pennation. Not all muscles have sarcomeres aligned along the long axis of the muscle. Pennate muscles have muscle fibers that align obliquely with the tendon, and the angle of pennation with the tendon can affect the number of sarcomeres per cross-sectional area, and thus maximal force capabilities. Any factor that affects angle of pennation would thus affect strength and velocity of shortening. As long as the cross-sectional area remains the same, muscles with greater pennation, muscle fibers at a greater angle from parallel to the tendon, have more sarcomeres in parallel and fewer sarcomeres in series. They are therefore better able to generate force, but have a lower maximal shortening velocity than non-pennate muscles. Angle of pennation may vary depending on hereditary factors and even training, which could help account for some of the differences in strength and speed seen in individuals who have muscles of the same size. Sarcomere and muscle length. The number of myosin cross-bridge heads that can align with active sites on the actin filament at any given time depends on the relative length of the sarcomere, or the relative length, percentage of contraction, of the muscle. When a sarcomere is very short, potential maximal force is reduced because actin filaments overlap, reducing the active actin sites that are available for myosin cross-bridge contact. When a sarcomere is relatively long, the potential maximal force is also reduced because potential actin sites are beyond the reach of the myosin cross-bridge heads. When the sarcomere is at its approximate resting length, however, the optimal number of myosin cross-bridge heads can align with the active actin sites, and maximal force potential is greatest for that muscle. This means the amount of force that a muscle can exert is related to its length. Peak force production is usually seen at resting length or slightly greater than resting length. This is known as the length-tension relationship of muscle. Pre-stretching. Pre-stretching a muscle just prior to concentric action can enhance force production during the subsequent contraction. The increase in force production is called stretch shortening potential, or more commonly, the stretch shortening cycle. This enhancement is probably caused by the combined effects and use of elastic energy in the muscle, primarily from the stretch of the myosin cross bridges, and stretch reflex potential, activation of myitic stretch reflex caused by the rapid stretch of muscle. The stretch shortening cycle is a very important phenomenon in sport, since it occurs so frequently, and it is an integral component in such activities as running, jumping, throwing, and striking. Stretching a two-joint muscle at one joint may increase the muscle's ability to generate force at the other joint. For example, the hamstring is stronger and uses less energy during knee flexion when the hip flexed than with the hip extended. The improved function of the hamstring seems to be related not only to the length-tension relationship of the hamstring, but also the use of elastic energy in the stretched muscle. It appears that muscle may use elastic energy gained even from the static stretch. Exercise-induced muscle damage. Hard exercise can induce delayed onset muscle soreness, DOMS, which normally occurs 24 to 72 hours after exercise. What causes DOMS is not known definitively. However, exercise-induced muscle damage followed by an inflammation-induced increase in fluid in the muscle probably causes the muscle discomfort, at least in part. Eccentric muscle accidents induce the most DOMS. DOMS is normally associated with a reduced in muscle strength, and following long-duration aerobic running may impair the ability of the muscle to generate ATP from oxidative processes for several weeks. As would be expected with reductions in strength and aerobic ATP generation abnormalities, DOMS has been found to be associated with abnormalities in sarcoplasmic reticulum and degradation of titin filaments, disruption of Z lines, and mitochondrial swelling. Older muscle. Muscle function is reduced in older adults. The ability to do daily tasks is a large problem in geriatric populations. Skeletal muscle, sarcopenia, is reduced muscle size and strength. As a result of aging or inactivity is amplified in weight-bearing extension muscles. As a result, loss of function appears most pronounced in lower limb and trunk extensors, followed by lower limb flexors, and last, upper limb extensors and flexors. Further, muscle atrophy with aging results from the loss of both number and size of muscle fibers. Type 2 fibers appear to be the most affected than type 1. Resistance training readily increases muscle mass and strength in older adults, suggesting that inactivity plays an important role in the progression of age-associated sarcopenia. Muscle fiber type. Fast twitch or type 2 muscle fibers may be capable of faster and stronger contractions, but may be less capable of generating energy through oxidative processes and less efficient in use of ATP. Thus, individuals with high proportion of fast twitch fibers may have an advantage in explosive short-duration events, but a disadvantage in long-duration endurance events that require both aerobic endurance and muscle efficiency. Fast twitch muscles fibers also seem to be more easily injured during high-intensity exercise, suggesting, at least in theory, that power athletes may be more vulnerable to DOMS. Conclusion. Strength conditioning professionals need to be aware that many factors affect contraction of individual muscle fibers. Several questions should be answered before a sport-specific training program can be developed. What is the speed of the muscle action? Is the muscle concentric, isometric, or eccentric? Is the muscle stretched prior to action? Through what range of motion does action occur? Is the muscle preloaded prior to the action? Answering these questions should help strength conditioning professionals understand how the muscle functions in any sport and allow them to develop programs that will assist an athlete in obtaining specific training adaptations.